Yes, dear students, in continuation with the previous lecture, today we are going to discuss about the remaining drugs from the anti-malarial class. So the remaining drug that we have unable to discuss last time, that is amodiaquine. Now, amodiaquine is a quinoline drug having a chloro group and is at its 7th position. So this is the structure containing having a quinoline nucleus and presence of chloro substitution at the 7th position, and as well as this drug is having a substitution of aryl amino group at 4th position. So this is a quinoline nucleus in which the substitution of chloro group is at 7th position and substitution of aryl amino functional group at the 4th position. This is going to have the similar mechanism of action as like that of chloroquine. Amodiaquine is having tend to be administered in the areas of where the chloroquine resistance is developed. So this is particularly used for those type of malaria in which we are unable to treat that case with a chloroquine drug molecule, and in that case only we will go for the treatment with the amodiaquine compound, while some patients are going to prefer its tendency to cause less itching than as compared to the chloroquine. Then it is used for the chloroquine resistance and for the treatment of infection if it is caused by the falciparum malaria. Now the next drug is primaquine phosphate. So the primaquine phosphate is actually the substituted nitrogen substituted amino derivative in which there is presence of pentane 1,4 diamine substitution and the presence of 6th methoxy substitution in the quinoline nucleus. Now this is particularly the N4,6 methoxy quinoline nucleus in which there is substitution of the side chain with the pentane 1,4 amine nucleus. This is most effective against the stage that is gametocytes. So this particular drug is going to be act at the gametocytal stage, but also acting on the hypnozoid stage. So the blood schizonticide and the dormant of Plasmodium vivax, Plasmodium oval are particularly treated. This type of infection of parasitic infection can be treated with the primaquine phosphate type of drug molecule. This is an anti-malarial prescription medicine and particularly approved by the US Food and Drug Administration. So this drug is particularly approved by US Food and Drug Administration Authority, and this is preventing the relapse of malaria which is caused by the Plasmodium species. So the mechanism of action is not fully understood about this drug molecule, but is thought that it will block the oxidative metabolism of the Plasmodium species and bind with the nucleoprotein drug molecules and interfering with the protein synthesis of the parasitic compound. Then it is also intercalate readily into the double-stranded DNA molecule of the parasite and inhibit both the DNA and RNA polymerase reactions of that Plasmodium species. That means they are particularly acting blocking the oxidative metabolism, as well as they interfere in the process of formation of that they are going to inhibit the DNA and RNA polymerase. So this is particularly the drug molecule that is primaquine. Our next drug molecule is pamaquine. Now pamaquine is acting as similar to that of the primaquine. It is used in the treatment of malaria which is caused by the Plasmodium vivax and Plasmodium ovalis species. Now this is also the drug molecule having similar adherence and looking similar, having structural similarities to that of the primaquine nucleus. This drug is also having a presence of 6th methoxy substitution in the quinoline nucleus, as well as the same drug molecule is also having a presence of 1,4-pentane diamine type of nucleus. So this drug is going to show same mechanism of action as like that of primaquine phosphate. The next drug molecule is quinacrine. Now quinacrine is actually an acridine derivative. Now this is actually and presence of acridine type of nuclear structure and having antimalarial action. Now this acridine class is going to have the action against Plasmodium species. Then the mechanism of action is they are going to intercalate or they are going to inhibit the DNA formation, DNA, and inhibit the succinate oxidation and interfering in the electron transport of the parasitic nucleus. Now this drug is particularly mainly used in the treatment of if the malaria is caused by the Plasmodium vivax and Plasmodium ovale type of species. This is also used in the giardiasis and giardia lumbalia type of infection. This is also used in the transcervically as a female sterilizing agent. So these are some of the applications of the drug molecule that is quinacrine nucleus. Now this particularly have the presence of methoxyacridine, presence of 6th chloro which is required for the antimalarial action, and this is acridine nucleus in which it is substituted at diethyl pentane 1,4-diamine type of nucleus in the middle ring. Our next drug molecule is mefloquine, in which the mefloquine bears some degree of structural similarity with the quinine type of structure. Chemically, it is 4-quinoline carbinolamine. It is marketed in the form of racemic mixture. This drug is particularly marketed in the form of racemic mixture, where the erythroform is more active than the threoform. So this drug is particularly marketed in the form of racemic mixture, and the racemic mixture where the threoform is more active than the threoform. This is orally active derivative of 4-quinoline. This drug is particularly orally active. Which drug? That is mefloquine is orally active, derived from the 4-quinoline methanol. About 95 to 98% administered dose is bound to the plasma protein of our body. This drug is particularly having plasma life up to 17 days. Principal metabolites of this particular drug molecule include 2,8-bis-trifluoromethylquinoline. This particular drug is excreted in the form of feces. So the this drug is excreted through feces. The mechanism of action of this drug is predicted that this drug is acting at erythrocytic stage in the life cycle of Plasmodium. So this drug is particularly acting on the erythrocytic stage. This is a very potent blood schizontecide with a long half-life up to 17 days. This is thought to act by forming a toxic heme complex that yesterday we have talked, that is they are going to form the toxic heme complex when they are going to be act with the hemoglobin of our body. They damage the parasitic food vacuoles and interfere the ability to metabolize the erythrocyte hemoglobin. It affects the ring stage of Plasmodium falciparum and Plasmodium vivax by inducing some of the morphological changes. So they are used in the treatment of both infections, that is Plasmodium falciparum and Plasmodium vivax, Plasmodium ovale, Plasmodium malariae. So this is now used in the prevention of resistant strains of all the 4 species of malaria. This is effective in the prophylaxis therapy for it is now strictly used for the resistant strain and usually combined with the artesunate drug molecule. Our next drug molecule is biguanides class, which is having an application as an anti-malarial agent. Now what are actually these biguanides? Biguanides is a large number of biguanides and dihydrotriazines have been synthesized and tested for their anti-malarial activity. Biguanides are largely having a pro-drug type of actions and they are not active until they are metabolized in vivo into the dihydrotiazine type of metabolite. The first drug is proguanil. Proguanil is a biguanide having synthetic derivative of pyrimidine. So this is a synthetic derivative of pyrimidine having a mechanism of action, but primarily it is mediated through the conversion of active metabolite. Just now we have talked about that these drugs are actually belongs to the class of prodrug species. This proguanil is first synthesized in Britain in 1945. This proguanil as a prodrug is first of all synthesized in Britain in the year 1945, and the next drug molecule is cycloguanil. Cycloguanil is metabolized to the active triazine nucleus. Cycloguanil is responsible for its anti-malarial activity. But both of these drugs are prodrug, and these prodrug first of all in vivo converted into the triazine type of active metabolite in our body. The antimalarial activity of cycloguanil is due to its structural and functional similarity with the resistant to the action of pyrimethamine. So this drug is going to have a structural similarity with the pyrimethamine type of anti-malarial agent, and due to this similarity the parasites that are resistant to the pyrimethamine also exhibit resistance to the action of this drug molecule. It inhibits the dihydrofolate reductase enzyme and interfere in the folic acid metabolism. This particular process leads to the inhibition of nuclear division in the malarial parasite. That means this prodrug is acting on the conversion and formation of the folic acid. Folic acid procedure involves conversion of dihydrofolate into the tetrahydrofolate, and this particular step is inhibited by the drug molecule that is cycloguanil pamoate, which is a prodrug, and due to this they are going to inhibit the nuclear division of malarial parasite. The cycloguanil has a duration of action up to several weeks because this is a prodrug. This is going to have its action after several weeks. The drug damage gametocytes so that they fail to complete their cycle in the mosquitoes. So this is having a very particular mechanism of action. They are going to inhibit conversion of formation of folic acid, conversion of dihydrofolate into tetrafilate, as well as having a long lifespan in the body up to 14 days. The drug as well damage the gametophytes. Cycloguanil acts slowly and effective away against susceptible strains of the Plasmodium. The development of the drug resistance limits its clinical use in the oral formulation. So this is going to develop a resistance, so they are less effective orally, while in the form of it is used intramuscular injection and provides longer duration of action. Structure activity relationship studies shows that the importance and presence of halogen atom in the phenyl ring gives an additional activity towards the malarial parasite. So this is all about the bigonides and dihydrotriazine type of nucleus used in the treatment of malarial parasite. Some of the other derivatives, that is pyrimethamine, which is used as a anti-malarial agent. Now this is coming under the classic class of pyrimidine analogs, which are used as an anti-malarial agent. Particularly pyrimethamine is a synthetic derivative. This is a synthetic drug derivative of ethyl pyrimidine with potent anti-malarial action. Pyrimethamine, a diamminopyrimidine analog. This drug is going to have the presence of diaminopyrimidine analog, and this is a anti-parasitic agent used to prevent and cure the malaria. Pyrimethamine also used in the treatment of adjuvant in the treatment of uncomplicated chloroquine resistance. So this drug can be used, we can use this drug when the chloroquine resistance is developed. It acts by, what is the mechanism of action? So this drug particularly acts by inhibiting the dihydrofolate reductase. Now this drug is particularly acting via inhibiting the dihydrofolate reductase in the parasite and preventing the biosynthesis of purines and pyrimidine. So thereby process of DNA replication is inhibited, cell division and reproduction is inhibited in the malarial parasite. They also act primarily on the schizones, and they also act primarily on the schizones during the erythrocytic phase. Nowadays it is only used in the concern with the sulfonamide. So they are used or they are combined with the sulfonamide class of the drug. It is used in the suppression of chemoprophylaxis of the malaria. They are also used along with the sulfazine and azithromycin for the treatment of toxoplasmosis. The last drug molecule, that is artesunate, which is used as a anti-malarial agent having some of the complicated structure. Now this artemisinin is derived from the leaves and flowers of, this particular drug is derived from the leaves and flowers of annual mugwort, that is Artemisia annua, and has been used in the traditional Chinese medicine for centuries ago. Artemisinin and its semisynthetic derivatives, collectively known as artesunates, and this is artemether. They are used to treat the tropical infectious disease of malaria, especially in the treatment of uncomplicated Plasmodium falciparum malaria. So this particular drug is used in the treatment of uncomplicated Plasmodium falciparum malaria. And the last drug is artemether. Artemether is an anti-malarial agent which is used in the treatment of malaria, which is particularly the 10 pan-methoxy 369 trimethyl decahydro 312 epoxy diazepinoisochromene type of nucleus. This particular drug is also derived from the natural source, that is Artemisia annua, used in the Chinese traditional medicine, that is quingosu. This drug is particularly containing the presence of sesquiterpene lactone from the terpenoid class with a chemically rare peroxide beige which is linked to the endoperoxide beige. This drug is active against multi-drug resistance, that is Plasmodium falciparum. This is also acting with a heme in the parasite and finally lead to inhibit the protein synthesis and ultimately results in the lysis of parasite cells and also arrest the cell cycle of malaria. That means this particular drug is going to arrest the cell cycle of the malaria. So used for the suppression of chemoprophylaxis of malaria. Last drug is atovaquone. Atovaquone blocks the mitochondrial electron transport system at complex 3 of respiratory chain of the protozoal cells. So thereby they inhibit the pyrimidine synthesis and preventing the nucleic acid and DNA synthesis in the parasite or malarial parasite. This drug is also having a conjugated naphthalene structure. The naphthalene nucleus is substituted with the chlorophenyl cyclohexyl 3-hydroxynaphthalene 1,4-dione. This is used in the suppression and chemoprophylaxis of malaria and also having anti-pneumocystic activity. So this particular drug is going to have the anti-pneumocystic activity against the malaria. So this is all about malaria and the drug used for the treatment of malarial parasites caused by 4 particular species: Plasmodium vivax, Plasmodium malariae, Plasmodium ovale and Plasmodium falciparum. Thank you. Soon meet in the next lecture. Thank you all. Please do subscribe to the channel so you will notified for the upcoming lectures of the particular channel, that is pharma chemistry altogether. Thank you.